giving me the opportunity to spend this day. And I have a message. I think I clicked it away. Yes. And so, yeah, I will talk about the role of biostatistics in pharmaceutical drug development. And the way how I'm going to do this is introduce drug development. And then you will see drug development is a very lengthy and complex process. And I will give examples at various stages throughout drug development where biostatisticians can contribute in a meaningful way. So with that, the motivation. Why do we think is biostatistics at all needed in drug development? So you may have heard about the thalidomide disaster 50 years ago. In the 1950s, it was introduced and developed as a sleeping pill. The problem being is that it was quickly discovered to help pregnant women to relieve morning sickness. And as a consequence, in the 1950s and 1960s, more than 10,000 children were born with deformities as a consequence of thalidomide use. The problem at that time being is that there were very few toxicity studies as part of the regular drug development program and essentially no statistics. These days, the US FDA, for example, has more than 400 statisticians across different divisions, drug development and drugs and biologics and radiological devices and health, but also tobacco and cosmetics. But altogether, there are more than 400 statisticians or close to 400 statisticians as far as I know. At that time, probably there was less than a handful of statisticians at FDA. So a lot has changed since those days, not only in terms of statistics, but the whole drug development process has completely changed as a consequence of that disaster. And one particular consequence is that statistics plays a major role. But it also indicates that drug development is a very lengthy process. So from the time it takes from the very first time a molecule is being synthesized all the way it becomes on the market, it could easily be 11 to 15 years. That's a huge and very lengthy process, right? And these are some figures that you can find in the internet, but what people say is that if you have 10,000 compounds, chemical molecules being synthesized, only one makes it to the market. And you see the different phases of drug development, and actually drug discovery and drug development. So we say the initial part, where you're looking at the molecules, we call that preclinical phase or research. So this is where you're developing the chemical entity, the molecule. You're going into animal tests and in vitro, in vivo assays. But only once you start in humans, that is when you would start with the clinical phases. So these would all clinical phase 1, 2, and 3. These would be the trials in patients or in healthy volunteers in humans, so to speak. And even that part, that's the longest part in humans. I think the research part may take about 5 years, and then you have another 10 years, say, in humans, until the drug is being accepted and approved for use for patients in need. And you see the differentiation, clinical phase 1, 2, and 3. Clinical phase 1, this is first in human studies, very small studies, and often in healthy volunteers rather than patients. So this is really very small studies just to see whether there are any immediate toxicities and tolerability issues. And if these have been clarified or cleared, then you go into phase 2, which is more to learn about the effects of the drug. So this is about the learning phase. And typically this would be conducted in patients. And once you have learned about the dose, the right population, and the right formulation of your therapy, then you go into phase 3. These are the large confirmatory trials. So this is where you would have those who attended my previous presentation, where you would have large independent phase 3 trials and if those are positive, then you go to the FDA or EMA here in Europe for registration. So drug development follows a very well-defined path. It's a sequence of many, many trials and experiments. I went through all this on the previous slide in a visual way, but you see it's a very complex trial. It's a very complex path with a high risk to fail, right? So even on the previous slide, you see from 10 compounds that go into the clinical phase, only one may make it to the market. And you also see the high associated costs. So I think even up to phase 3 programs, I think every second phase 3 program fails. And these programs are very expensive. They cost millions and millions of dollars. Okay. So this is why having innovative trial designs is so important. Innovative analysis approaches is so important to increase efficiency and support making good decisions, so that you can really reduce this high risk of failure. So what I'm going to do is then now go through the different phases and quickly show you a few opportunities for biostatisticians to contribute, starting with drug discovery and preclinical phases. So a classical drug, the type of tablets that we're all used to, is a simple chemical formula, as you see it, for example, here, fingolimod, which is an oral treatment for multiple sclerosis, developed by my company, the first oral treatment for multiple sclerosis ever. And but we also have other types of medicines like biologics. These are huge, big antibodies. This is not just simple chemical formulas, but a classical drug would be just such a simple chemical formula. In the past, drugs would be discovered by isolating, for example, an active compound from some herbs or remedies or other remedies. And Artemisia is a perfect example where in China they were using it for centuries against, for well-being. And it was in the 1970s then being extracted, the active ingredient. And these days it's a standard treatment for malaria. However, the knowledge about disease, genes, and human body is increasing, but still very limited. So this is not the standard way of how we do develop drugs, but the reality is that we use high throughput screenings. So we use some sort of robotics where we quickly scan 10,000, 100,000 of chemical compounds that are, automatically designed. There's no human who's designing those chemicals anymore. And the objective is just to see whether there's any hit to any chemical reaction, so to speak. It has nothing to do with the drug at that point. There's no animal and no human. It's completely automized. But you can imagine there are huge statistical challenges. Very often we have not even a single replication. We have very highly variable assays and we have large number of tests. And still you need to understand what is a hit and what is not a hit. So clearly you have huge statistical problems in this area. So this is in the labs, but before we go into humans, then we would have what we call preclinical development. So after a new compound is being selected, further tests are being performed mostly in either in form of in vitro or in vivo studies. In vitro means that you have isolated cells, at least you have some biological mechanism, or even in animals. So this would be in vivo studies, and often we use rodents, as you can see on the pictures here, with the guinea pigs and the mice. So toxicity studies are key focus of preclinical studies, of preclinical development, and it still may take several years. So the National Toxicology Program in the US has really a very standardized 2-year program for animal toxicity studies to see whether any tumor is being developed, and if you give that compound, this new drug, potentially new drug to mice or to rodents. So even that takes quite a bit of time, and you can imagine the importance of that if you go back to a preclinical study to investigate the effect of thalidomide. This is this active compound that I mentioned before from the disaster from the 1950s. So here they investigated the effect of thalidomide at different doses. You see 550 and 500 mg. You see the range goes from 5 to 500, so it's a 100-fold range. And they investigated the liter weight at the different doses. And the liter weight does depend on the gestation time. It doesn't depend on the liter size. And the objective was to show whether thalidomide has any toxic effect. And here are the results. And you see that the weight, you see that here. It's 32, 29, 29, 29 with some standard deviations, and you also see the impact on the gestation time and liter size. And the question is, if you see such a result, is lower weight is a sign of a toxic effect. And the question is, does it mean that a weight decrease with larger dose? and you want to formally test for that. So this is a trend test that they often use in preclinical assays like this. And here you see the graph. And I think if you look at the graph, it becomes immediately clear that even with the lowest dose, you already have a substantial reduction in the litter weight. And indeed, the p-value is highly significant. So this shows if such a study would have been done in the 1950s, very likely thalidomide would not even have come to the market, not even like in the clinical phase. And this is why such trials are important, and also why you need statistical input in the preclinical phase. So assuming that you develop your drug, you have your in vitro and in vivo tests completed successfully, then you go into the clinical phase. And this would include a number of different trials at the very early stages. First studies of drug in humans, so first in human trials. And essentially what you would like to understand is the pharmacokinetics, the pharmacodynamics of the drug, and I will explain what that means. And as a very first objective, you would like to understand what's the maximum tolerable dose. So what is the maximum dose that a human can still tolerate the drug? It's not yet safe, but at least you don't have an immediate toxic reaction, so to speak. So that's all is a typical objective of clinical phase 1. And as I said, in the and you see it in the first row, first in human studies are typically very small in sample size because you don't want to, have a too large risk and really just expose a very few humans to this very new compounds. The studies typically last several weeks, maybe months, and very often these are healthy volunteer studies. Excuse me. So these are not patients, but these are healthy humans who voluntarily join a clinical trial, and really the dose that being administered typically very small. And as I said, they want you would like to understand just the chemics of the compound, like the pharmacokinetics, pharmacodynamics, bioavailability, just to understand how the compound is being absorbed by the body, and what the compound does to the body. So pharmacokinetics and pharmacodynamics is very important in these early phases. So pharmacokinetics explores what happens to a drug in the human body, so how it's being processed. And so they talk typically about absorption, distribution, metabolism, excretion. I'm not going through the individual terms, but it's this ADME framework that they use in this context, as opposed to pharmacodynamics, which describes the effect of the drug on the human body. So how the drug interacts with enzymes and other proteins, binds to receptors and chemical reactions. So all this is taking place to understand what the drug, how it is interacting in the human body. Now, what is being then done is then you try to model that somehow, and a very common class of approaches are so-called compartment models. And here's a very simple one compartment model, is the most simple one. You take a tablet, so it's orally administered, and you see it in the left-hand side picture, how the dose goes to your stomach, it's being absorbed in the blood, and then over time is being eliminated from the body. So that gives you, this is the one compartment, so to speak, and you have a certain absorption rate, you have an elimination rate, and on the right-hand side you see the red and the blue lines, the absorptions and elimination of the drug, and the black line is essentially the difference of those two curves. And so the black line is the drug remaining in the blood. So this would be this typical pharmacological curve. You take the drug, it's being absorbed, and then over time is being eliminated from that. And it's the difference of these two curves. And very often this is solved by using differential equations, as you see it here below. So this is a simple pharmacokinetic model, and many more complex models can be thought of and need to be estimated. The parameters need to be estimated based on the data. Another objective is beyond understanding what the compound does to the body, or also what the body does to the compound. Another objective beyond this more chemical scientific discussions is also what is the smallest dose for which a certain percentage of subjects do not tolerate the compound. So we would like to understand what is like the minimum, the maximum tolerable dose, and this is the MTD. So we feel that any dose smaller than the MTD could be explored in the further exploratory trials in phase 2. And this is typically based in oncology. You will, it's a very common approach to really escalate the dose until you have identified this MTD, and this escalation is based on well-defined what is called dose-limiting toxicities. So these are events that patients could have, and that would be associated with the toxicity of the treatment. And what you would like to do is then, the probability of a patient to experience such an event, such a dose-limiting toxicity, this probability should be bounded, for example, by 33%. And then if you have such a, on the x-axis, the dose levels, on the y-axis, the probability of such a DLT, then, and you have somehow a dose response curves as through the solid black line, you're trying to estimate what is this dose associated with that value on the y-axis. So you're trying to estimate here the value here on the x-axis, which is your maximum tolerable dose. So you have this dose toxicity curve, which is the functional relationship between the dose and the probability of having a DLT. And the targeted toxicity level then is defined as the probability of getting such a DLT, as I mentioned. And very often we use the threshold 0.33, but obviously that's an arbitrary threshold. It could also be 0.2, it could be 0.5, but yeah, you see how it's being interpreted. And what is then often done are done so-called phase 1 dose escalation trials. So these is one form of so-called quote-unquote adaptive trial designs, where you would run the trial in groups of patients, a very small number of patients, of cohorts of patients, like 6 patients, and then you will increase the dose with each group until you come to this toxicity problem. And once you're getting closer to observing more and more toxicity, that is DLT events. And these days, since the early 1990s, we are using many different forms of Bayesian adaptive designs, where based on the current data, we decide on what the next dose could be. And obviously there's substantial uncertainty during and at the end of the trial, and statistics plays a major role here in designing such trials, in analyzing such trials, and to also help during the trial guide decision makers under this uncertainty. So statisticians play a very important and vital role here. Now this would be the phase 1 trials, and moving forward, we would then consider so-called phase 2 trials, where we assume that the drug is at least not toxic, and we have understood the MTD, or what is like the largest dose that we can administer to patients. And in oncology, this highest dose is probably the one that we would then investigate further, because it's a lethal disease, and you don't want to go with a lower dose. But in non-oncology drugs indications, you may not want to go with those higher doses. Maybe a lower dose would be equally efficacious, but have less side effects. So what you really would like to do in non-oncology settings is first to understand whether the drug has any efficacy at all. So far, you see the phase 1 was mostly about the pharmacokinetics and the toxicity. Now is the first time in phase 2 we will look at the efficacy. So whether there's any evidence of the drug actually doing something useful to your drug, to your body, to your disease. And dose finding becomes a very important aspect. So what is the right dose for the right patients? So this would be the clinical phase 2 exploratory trials or learning phase. And these trials will be a bit larger than the phase 1 trials. So here we talk about, say, from 50 to 200 patients in phase 2. These trials will typically take several months, maybe up to 1 year. And you will definitely have patients rather than healthy volunteers, because we are looking at efficacy. So we want to see a proof of concept whether the drug actually helps the patients and really addresses is a treatment for your disease. And so this would be the proof of concept step to understand whether you have any sort of efficacy signal. And we would like to understand what's the dose-response relationship, and we might have other exploratory studies, for example, looking into biomarkers, subpopulations. All this is part of your phase 2 program. So how does such a proof of concept study look like? So this would be the first study as part of your formal phase 2 program. We call it phase 2A trial. So this is the first time outside oncology where you are testing the drug in patients for efficacy over placebo. And the objective is that you have enough evidence that your drug has a positive effect in patients. And then you have certain proof of concept criteria to guide your decision. So it will not be a regular t-test, so to speak, and your p-value is less than 0.05, but rather we have different thresholds and we calculate Bayesian probabilities to understand whether these thresholds are being matched or achieved or not. So for example, we would have MD, that's the minimum difference, and TD would be the target difference. So these would be two different thresholds. If you're below the minimum difference, then you are saying that your drug is really not efficacious at all and you don't want to go ahead. But if you're achieving even your target difference, then you have a strong belief that your drug is really very effective and you definitely want to go ahead and continue developing your drug. If you're somewhere in between, this is the yellow, that's the yellow part, means, well, my drug is somehow effective, but it's maybe not super effective. So maybe I need to collect more data before I move on with my actual development program. That's in a nutshell a little bit how this bottom figure has to be interpreted. And what is being then done is use Bayesian proof of concept decision criteria. And you know, if you have zeta being the treatment effect that you're interested in, well, then you're interested in what's the probability of that zeta to be less than your minimum effect or larger than your target effect, given your data. And then you have certain thresholds that you want to meet. So in red, you see this 0.05. So it's to match our typical type, significance level of 5%. And, in green, you see the other threshold of 0.5. And obviously all these thresholds, they are just somewhat arbitrary. But what you really would like to understand is that you don't develop a drug which does not even meet your minimum difference. And ideally, you would like to develop a drug that is, where you achieve with a high probability your target difference. And we tend to use Bayesian probability, posterior probabilities for that. So these are the concepts of proof of concept. This is the idea of proof of concept trials. And if you feel that your compound does work and it's worth to be developed further, one typical trial is so-called dose-finding trials. And to motivate, I'm using here a quote from Paracelsus from 500 years ago, who was a remarkable medicine, like a very modern type thinking person. I think he was revolutionizing medicine at that time, 500 years ago. He already had this quote and realized that all things are poison and nothing is without a poison. Only the dose permits something not to be a poison. So, I mean, if you think about drinking water, it's obviously nothing harmful unless I drink 10 liters of water within 24 hours, I will die. And like table salt, if you eat 500 grams of table salt, I'm sure you will at least have to go to the hospital. Maybe you will die as well. So it's all about the dose. And likewise, thalidomide, if you use thalidomide at a very, very, very low concentration, obviously you probably won't have side effects. So though he was very clear, you need to know the dose 500 years ago. And so it's really modern thinking. And but using our today's terminology, you need to establish dose response, both for efficacy, but also for safety. So is there any evidence of a dose-related treatment effect on both scales, efficacy and safety? Which doses are different from your placebo control, for example? How does your dose response shape look like? It may be a nonlinear regression model that you want to fit. What is your optimal dose? How do you define optimal dose and how can you estimate it? So do you see there are many, many interesting statistical questions soon coming up. And characterizing the dose response for safety and efficacy is really critical, because if your dose is too high, you bring a compound on the market at a too high dose, then many patients will have tolerability problems. But if you choose a dose too low, then it is not sufficiently efficacious. Maybe you even not make it to the market because you feel as a company, it's inadequate, the efficacy is not adequate. So finding that balance between safety and efficacy extremely important for any tablet and any medicine that you will find that you can get in the pharmacy. And so and it's not an easy problem to estimate the right dose, because you cannot even tell exactly. Many cases, in many instances, you even cannot tell in a formal mathematical way what you mean by optimal dose. So this is the phase 2 trial. And as I said, in a phase 2 trial, we might be doing many other types of trials as well, like biomarkers, subgroup effects, and so on, to identify what's the right endpoint, what's the right population, and all this. And once you have, learned enough about your drug, then you are ready to embark on the big phase 3 trials, the type that I talked in the seminar before. These phase 3 trials serve for registrations, that is for going to the health authorities and tell, hey, I have a drug and I would like to get it on the market. And so I'm preparing myself for a submission. These trials can sometimes be huge. So I have seen mega trials, well, I haven't seen, but our company runs sometimes mega trials with 10,000 of patients. In the biggest trials, probably 20,000 patients, right? It's huge trials. And you can easily imagine they take years. And obviously these are patients, but they are important for your submission. So because they are so expensive and they are so huge and they are so important, statistics plays a major role. Every single trial in this domain will be very well designed, and we have lots of statisticians talking here. So, and in my organization, in my company, we probably have 500 statisticians, pharmacometricians globally. Out of these 400 or 500, I'm sure there's more than 300, like about half of them, or maybe 300, who are working on typically on phase 3 trials. I think that's probably a fair assessment. So what do we want to do? Well, clinical phase 3 studies include usually 1 or at most 2 doses of a test drug, because you already have done, hopefully, your dose finding trials before. And you will have compare your drug versus an active comparator or placebo. And typically you will have several hundred up to several thousands of patients being included. And the effect of the drug has to be shown using a recognized clinical endpoint. It will have to be a formal hypothesis test at the 2.5% one-sided significance levels. And what I said in the previous seminar, US regulators will require statistical proof of efficacy from 2 independent trials. So you might still run additional studies, or maybe you have expansion cohorts to investigate more the safety database so that regulators have enough safety data. If your efficacy studies do not have enough patients or not long enough. So there might be some additional studies in that domain just to make sure that you have enough safety data available for regulators to make their decisions. So, and on the next few slides, I will then highlight just a small selection of items that statisticians in drug development will typically be focused, they will be facing in our standard phase 3 trials. And a common problem is obviously, can we have faster trials and programs? And adaptive designs is a very popular way of thinking to reduce the sample size, because you're trying to react to the results of an ongoing trial and be adaptive. To explain that, maybe I should say that the traditional phase 3 trials, you would not be allowed to observe any data from patients during the trial. Because once we call it unblind the patients, once you get to see the results of patient data during the trial, it could introduce some sort of a bias for the remaining conduct of your trial, and regulators will never allow a company to see data from an ongoing trial. So typically we call that being blinded. So the company or any trial management personnel will have to be blinded with respect to the treatment code. But there might be possibilities to run adaptive designs. I already mentioned these dose escalation studies in the phase 1 world. You can run adaptive phase 2 dose finding trials And occasionally, you could also run an adaptive design in phase 3. The idea is that whenever you have a new cohort of patients coming in, you randomize them, you observe the data, and then you decide whether you want to stop the trial early, you initiate some sort of a next trial or some other decision process, or you continue in the ongoing trial, and the next cohort of patients will then be randomized again based on your updated information. So this is possible to some extent in phase 3 trials as well, but the type of adaptive designs we use in phase 1, phase 2, and phase 3, and all different types of adaptive designs using different methodologies, different designs. So adaptive design is really a big umbrella term. In addition, we might look into efficient, complex, innovative designs, master protocol designs, if you have heard about these terms. So this is just the next level of adaptive designs. But sometimes you might even consider trying to get conditional approval based on real-world evidence. So that maybe in the phase 3, we have not yet completed our phase 3 program, but maybe sometimes you have already the drug for a different indication already on the market, and there might, what we call real-world evidence is through registries or other databases, there might already be some information which could help us to get some conditional approval for our drug, and then we have to complement it, by running an expensive phase 3 trial to get full approval. So you see there are different ways and strategies how you can speed up the individual trials or entire programs based on some of the ideas laid out on this slide, and statisticians will play an important. Another element is how you characterize treatment effects. And those of you who have heard about the estimate framework may recognize this picture, but essentially you have to be very clear what is the treatment effect that you're trying to estimate. And what you see here on this picture is, so you have patients who are randomized to either standard of care at the bottom or to the new drug on top of standard of care at the top. But then you also see that some patients may have some events occurring during the journey within the trial. So for example, patient number 1 would stop treatment, end of treatment, and start a new therapy. Patient number 2 has end of therapy, but then withdraws the consent and essentially drops out of the study. Patient number 3 dies, and patient number 4 has no such post-randomization event. So the question is here, what is actually your treatment effect and how you take such post-randomization event into account when you think about the causal treatment effect? So causal influence plays into this estimate thinking process, and it's a more recent topic that came up in our world just over the last, 3 to 5 years, but it's a hot topic currently. How you describe the treatment effect you're interested in. But characterizing treatment effect is more than estimates. You can, for example, use some of the modern machine learning techniques to identify endotypes with best responses, or you can even develop more sensitive endpoints by a model. So there's quite a few opportunities, to get excited in the context of how you characterize treatment effects for our trials at the end of phase 3. For the purpose of registration and drug labeling, keep in mind that once we bring a drug on the market, it's not only about getting approval from FDA or EMA, the agencies, to get it approved, but it also has to be reimbursed. So there's a whole different set of agencies who will agree or disagree about the reimbursement. And obviously investigators, doctors, and patients will have to be informed, typically through the label, the package insert, what the drug does. And so this is where characterizing treatment effect has an immediate impact all the way to informing the patients. Now, so patient number 2, you see, stops treatment and drops out of the study. So you don't have any data for that patient number 2 at the end of the trial as you hoped for. And patient number 3 dies, and there can't be any data because he died. So this actually leads then to statistical problems such as missing data problems, very closely related to this estimate problem I mentioned on the previous slide. So missing data are data we intended to collect, but for one reason or another, we did not. And you see that in the picture, if you have a single patient over time, you keep observing the patient, but at some point, you see the shaded box, the patient drops out of the study or maybe dies. What you don't know what happens. Does he would follow the natural path of your curve? Would he follow the red curve or maybe the purple curve? You don't know. But you need to somehow impute the data under clinically plausible assumption. So how do you impute missing values? It's a big statistical problem. Multiplicity. I mentioned some multiplicity aspects in my previous presentation, but you can imagine the probability to commit at least one type 1 error increases with the number of hypotheses. You see that displayed on the right-hand side for hypotheses going up to 100 hypotheses. So if you have many exploratory endpoints, you will have to control for multiplicity because most likely, almost surely, you will have at least one p-value being less than 0.05 if you have many, many exploratory endpoints for efficacy. But also for safety, if you have many, many different adverse events, very likely you will see one adverse event being, like your drug being showing some signal for one type of adverse events, even if it was safe. So here the multiplicity problem is inverse. So you see a signal, but it shouldn't be there because your drug is safe. So adjusting for multiplicity has many different interesting aspects, and my previous seminar was going along these lines. The next one is we are running trials typically globally. So many trials are conducted not in UK or in Europe or in US, but many trials in phase 3 are conducted globally with centers in many different parts of the world. Actually, recruiting patients in Eastern Europe is much cheaper than recruiting patients in Western Europe, and likewise in the Southern Hemisphere versus the Northern Hemisphere. So companies like to recruit patients outside expensive US and European Union, but then you have the FDA and the EMA, they have to regulate drugs for their target population, which are patients in Europe and in US. So that obviously generates questions about regional consistency. And you see on the map, not so clear, but you see this is actually one person did it, looking at all the different sites, clinical hospitals that participate in one of our big phase 3 trials. And you see we have several sites in Australia, many sites in China, many sites in Europe, in Africa a few, in South America a few, and then several sites in the US. So you can imagine that the medical landscape in each region is different. Sometimes a certain co-medication is allowed, in other regions it is not, and then maybe the whole infrastructure, the medical infrastructure, the healthcare system can be completely different. So how do you make sure that you have a drug effect at the end, one drug effect? So you would like to show that you have somehow a regional consistency. The opposite problem is you believe that there is actually an effect in certain populations. So that's the opposite problem to what I just said. With our multi-regional trials, we would argue that we have a regional consistent treatment effect. Now you might want to expand into new populations. So you would like to extrapolate efficacy into neighboring population, like from adults to pediatrics. You would like to explore integrated data to discover new mechanisms, or use historical data as evidence instead of running a new trial. And what you see on the picture on the right-hand side, if you have this gray population, you would like to understand who are the responders, who are the non-responders, and who are the patients who react adversely to a drug. So this is all about actually identifying and explaining heterogeneous treatment effects, which is the opposite problem than we had on the previous slide. And evaluation of safety in phase 3. Most of what I just mentioned in the context of phase 3 trial is to understand and describe and characterize the efficacy. And this is where most statisticians would be focusing on, but each pharma company will have at least some group talking about safety evaluations. So the problem with drug safety is, like, for efficacy, you have a well-defined endpoint. Is it a reduction in blood pressure? Is it a reduction in HbA1c in a diabetes study? Is it the disability progression score in multiple sclerosis? You have a well-defined efficacy parameter. Drug safety is multidimensional because any adverse event will lead to some concerns about drug safety. And typically you cannot measure it by a single parameter. You might have a well-defined safety problem where elevating creatinine level in your kidney. So that's a well-defined safety parameter. But really, if you want to develop a drug and bring it on the market, you really would like to know that the drug is overall safe, right? So it won't be a single parameter. And a subjective expert review will have to be involved in the assessment of safety. So very often it's not just a quantitative safety assessment. Sometimes it is somehow subjective. And standard statistical tests are usually not performed, but may be conducted for specific adverse events. And safety information is collected from all trials and all phases and could be adverse events the way I just described, nausea, headache, and things like that, but it could also be common laboratory tests, vital signs, or ECG. So it could be different sources and different types of data that all contribute to your overall safety profile. And as a statistician, you will help the team to understand whether you have a safe drug or not. So this is all part of your phase 3. Once you have completed your phase 3, then you will continue and prepare for the submission. That is, have your big dossier that you will going to forward to the FDA. But even after the FDA or the agency has given market approval, we will still have additional studies being conducted. We call them post-marketing studies. These could be commitment to the agency. So maybe the agency says, yes, you can bring that drug on the market, but only if you do this additional study. Or we would like to expand the label. We would like to understand whether the drug works in additional populations, maybe in elderly patients, maybe in phase 3, or the drug label says, in all post-menopausal women until 65. So we would also like to understand whether it also works above 65, what happens in post-menopausal women. So you can immediately see how we have many ideas and efforts in conducting further trials after the drug being marketed. But one particular example I would like to show here is, first the basic characteristics. I think I mentioned all this, and I think I'm going to skip this slide. But these post-marketing studies are very broad. There are many, many different types of post-marketing studies. But one particular one is, sometimes a problem occurs with the drug being marketed because, some major safety signal comes up, and you need to run additional statistical analysis or even run additional trials to understand this safety signal being that is appearing. And so one is the Vioxx scandal from 20 years ago or 15 years ago, which made a lot of press, where the drug was approved in 1999, and over 80 million of patients had used the drug, but it had to be withdrawn 5 years later, 2004, because as part of some meta-analysis, clearly there was an increased risk of heart attack and stroke associated with a long-term high-dose use, which could not be detected, or at least it was not detected based on the phase 3 data. So a post-approval study of 36 months had shown that long-term use of the drug resulted in nearly twice the risk of suffering heart attack or stroke compared to patients receiving placebo. And you can imagine how this looks like if you have then many different types of studies. You see here 20 studies in this forest plot, and at the bottom, the combined relative risk was highly unfavorable against the drug. So you see the statistical discussions after marketing a trial could be to extend towards new labels, understanding the effect in other populations, but obviously you will have to sometimes be engaged with discussions like that up to the worst case, which is about removing a drug from the market, as in this case. So you have the whole bandwidth of activities even after the drug is on the market. So in summary and conclusions, drug development is a very complex process. I hope I could give you a quick glance, what drug development is about, and all different complexities. There's a very high risk to fail. The statistical challenges are enormous. At different phases, we will have different challenges, different levels of uncertainty, and therefore you will have to use very different methods at different drug development phases. But regardless, good statistical practice is badly needed, as it does lead to good decision making. And just the fact that FDA has close to 400 statisticians obviously speaks a very strong signal how important statistics is in drug development. Now, in my presentation, I was really focusing more on the classical role of biostatistics. What I have left out, but this is really, it's just coming up over the last 5 to 10 years, drug development landscape is constantly evolving. And these days, statisticians, and maybe beyond statistics, so-called quote unquote data scientists, with whom we are collaborating or we are ourselves turning into those type of activities, we have many additional ways of opportunities of contributing to drug development. And yeah, you see just, a couple of, cool pictures just to show that where, over the last 5 years, the scope of biostatistics has greatly expanded. And it's not just about the topics that I mentioned, but yeah, we have many different data sources these days, variable devices, streaming data, imaging data, genetic data, all this type of different types of data that in the past we would never, they were simply not available, or in case of genetic data, we would not collect those data, or at least the biostatistician would not have access to the data. But these days is greatly expanded scope for biostatisticians in drug development. So with that, thank you very much for your attention.
Thank you very much, Frank, for a really nice overview of the drug development process. So I'd like to open up for questions. So for those online, please raise your virtual hand. Alison can unmute you. And for those in the room, Alison have a roving mic. So do raise your hand and ask any questions. So while we're waiting for some questions, I'll start. So I guess one question I had was how you thought COVID-19 had changed any of this, or whether actually it's more kind of one-off unique event, whether potentially.
Yeah, that's a good question. I completely missed COVID-19 in my presentation, and it has a significant impact. Though I think, obviously it produces additional challenges because COVID-19 with all the lockdowns will impact all of our clinical trials. The hospitals are not going to admit certain patients anymore, and patients don't want to come to that anymore. So you have many sort of, obviously, clinical trial disruptions that in your regular ongoing trial. I'm not talking about developing COVID-19 treatments or vaccines. I'm just talking about the regular clinical trials. And as a statistician, it's just a mess of data that you now need to make sense of it. But in addition, I think there are also some, we will see how it works in future. But one element I clearly see is what we call decentralized trials, because patients would not come to the doctor anymore. So it's more and more in the last 2 years that patients would, if possible, get the endpoint being recorded remotely at home. So the sort of telehealth has become much more prominent these days. And I think as a company, most big pharma companies are now proactively thinking about how we can use telehealth, and call it decentralized trials as another term, how we can use these digital, whatever, collection of endpoint information at the patient's home rather than patients coming to us. And there are lots of additional statistical questions around this. So this is just one element which I'm foreseeing. I think those discussions are already taking place. Yeah.
Great, thanks. Alison.
So we have a question, with a hand raised from Stephen Coleman. Would you like to unmute yourself and ask your question, please?
Cool. Can I be heard now?
Yes, you can. Yeah.
Great stuff. Yeah. Now, thank you very much for that talk. I was curious about a couple of different things, particularly regarding the estimate of is it the MDT? Is the toxicity, the acceptable toxicity level? I imagine there's quite large uncertainty on that, or at least some. And I also imagine it depends very strongly, the estimate does, on the sample used in the phase 1 trial. So do you propagate that uncertainty through in the rest of the stages? And do you try and correct if you think your sample is unrepresentative of your full population that the drug will be used for?
Yeah, this is a great question. I think this is really a general problem, right? You have to start somewhere. And
Yeah.
you cannot, in the first in human trials or these dose escalation studies, already think about the population that you have in mind when it comes to marketing. Obviously, you can't, because you can only have 6 patients, so to speak. And so there's so much what you can do in terms of representation. So I think this is where I think statisticians can contribute, if possible, to quantify the uncertainty, if that was possible to quantify, but at least point out that if you have gone through this dose escalation study, that we can't. I mean, the point estimate in my picture, this one. So the point estimate in my picture here, I mean, this is really a point estimate with a huge confidence interval on the x-axis. And if you can quantify that uncertainty, that I think would already be a very important contribution as well. But just the fact that we have here a dose toxicity curve is already an improvement to how dose escalation studies were done, and maybe partially are still done, using so-called 3+3 designs, which have no statistical influence or probabilities at all. Here, we would just count how many dose DLTs we have, and based on the counts, then the medical people would escalate or up or down the level. So already having some sort of, for example, these posterior probabilities, that already is a step forward. But ideally, if we could quantify the uncertainty about the dose estimate, that would be even better. I don't know if that answers your question, but I think I'm just agreeing that this is not very representative at this point. Was that your question? Sorry.
Sorry, I was muted there again. Yeah, no, I think so. I think it's just that it's such a difficult problem, as you say. And just I'm sort of interested to see how you're currently dealing with it and how you hope to deal with it. So I think you did answer. Thank you.
Thank you. I think we have a few more online questions.
Yes, so Anna, you've got your hand raised. We'll just unmute you.
Hello, can you hear me?
Yes, we can.
Great, thank you. Thank you so much for the talk, Frank. It was brilliant. So I just had a quick question about the incorporation of genetic data that you mentioned briefly at the end. So is it still rare for genetic data to be incorporated in the drug development process, or is it now becoming more common? And also, would Novartis have a specific group of statisticians working specifically on the incorporation of the genetic data, or is that more general in Novartis?
Yeah, I think we are, no, it is still rare. I think especially in the later phases of drug development, maybe in earlier phases, especially in drug research, I think there we have actually such genetic groups, but we do use also I think we are now routinely collecting proteomics data as well. So yeah, I think because as a classical biostatistician, you would not even know what to do with those data, and you would not be even asked to look at those data. So in the research part of the organization, in the preclinical phase, we do have a dedicated group. In the clinical phase, I think we are still trying to understand how to best use such data. But yeah, it's becoming more and more prominent, that's for sure.
Okay, that's great. Thank you so much.
Any other questions? Yes, Lida.
Thank you for your talk. I really enjoyed it. So I think it was in the initial slides where you were speaking about pharmacokinetics and pharmacodynamics, and you spoke about the different models that are available yet to describe, for example, the absorption and so on. So I was wondering, are such models always available? And is it a problem if, what do you do if they're not available?
Yeah, no, that's a good question. And clearly this is out of my depth. That is not my area, but what I'm hearing is that no, in many cases, such models are not available, or they can become very complex because you have many different organs and you have how to model the interplay between the organs. I think it becomes very complex. And even if you could model it, maybe you wouldn't have closed form solutions as you have it here. So yeah, and that's where I think, the art of modeling becomes very important rather than having, like a simple one compartment. I think really becomes an art then, how you describe the pharmacokinetics in more complex systems. Yeah.
So how is there a way of dealing if you want to study a certain process, but there's no model available, or do you just avoid
Maybe both, right? I think, if you want to, take time off and really describe it and, go into system biology, I think there are ways how people try to overcome that problem. If you are in a hurry and you need to develop your drug, maybe you just use a simplified model just to move on with your drug development. So I think it's a combination of both, realistically speaking. Yeah, I don't know whether it partially answers your question, but yeah. But certainly content-wise, I'm not an expert at all. So yeah.
Could I quickly ask something else based on what you said about the simplified model? So then do you try and take into account that this is just a simplification, or do you just go?
Just apply it. I can just apply this simplified one compartment model and, assuming, I'm ignoring that in fact, complex biological mechanisms are much more complex. But clearly, we have a whole department who do this pharmacological modeling, so to speak. Yeah. So we have a dedicated group. Yeah. Yeah. And they have sometimes a few statisticians as well. So they have very mixed backgrounds. So this is not a biostat department per se, but we do have statisticians also in that like pharmacological modeling group. Actually, we have two of them. One in the early research area for when they are still and then we have one in the full development area. So we actually have two pharmacological modeling groups.
Nina.
So thank you for the great summary and the highlights on the main statistical problems. So I have a question related to the duality between theory and practice. And more specifically, in medicine and in clinical trials, there is a huge imbalance between theoretical works and what we actually in practice happens. So I wanted to know, is there an area among all these where statisticians can have more success proposing something, especially in the design part, and seeing actually their proposal then in practice? And yes, when I mean these parts, I mean like in a specific phase of a clinical trials. And since you mentioned also the adaptivity of clinical trials, since it interests all of these phases, I guess, especially 1 to 3, if there is a part where this adaptivity, the adaptive clinical trials can see light like in the future.
Yeah, no, these are great questions. So if I understood, there are two questions. Maybe I'll start with the latter one about the use of adaptive designs that I briefly mentioned. And I had shown these dose escalation studies for estimating the MTD, and that to me is one example of an adaptive design. You can also use that in the phase 2 dose finding trials. For example, you start with doses 10, 20, 40, 100 milligram. And then at the interim, at sometime during the trial, you decide to drop a dose, or maybe you add a dose. And that in phase 2 is particularly easy, and we call this adaptive dose finding trials, right? So it's quite possible. But here you would randomize patients to one of several doses at the same time, as opposed to the dose escalation studies where you would first wait for completing a cohort, and then you would decide whether to escalate the dose up or down or just keep the dose. So this is like more sequential, when those phase 2 would be like randomized in parallel to any of the doses, but then you can drop those, add doses. In phase 3, you can also run adaptive designs by, well, maybe you only have 2 doses and you drop 1 dose. It doesn't sound like big adaptation, but given that you may be looking at 1,000 patients, and if you drop 1 dose, then already you're saving several hundred patients to the wrong dose, so to speak. You could also, like, for example, start with an overall population, and then at some point, just reduce the randomization to a subpopulation. If you feel that you're having new information, you don't want to randomize the overall population, but just focus on the treatment effect in the subpopulation. And then all these master protocol designs, they come also across, phase 1, 2, 3. At every stage of drug development, I think people are suggesting the use, or at least investigating the use of quote unquote master protocol designs, which is a big term. So yeah, I think adaptive designs can be used at different, essentially at all stages of clinical drug development, although with very different techniques and under different requirements. And your question about where statisticians can best, if I understood it, best impact.
So it seems that there may exist already some techniques, of course, like, I don't
Yeah.
know, face for those finding doses, they are somehow adaptive by nature, right? So, and there exists some methodologies, and it seems that some of these methodologies are like the standards.
Yes.
So if a statistician would like to propose something new, for example,
Yes.
how high would be the resistance coming from,
Yeah.
I don't know, regulatory agencies
Yeah.
in terms of that? Or should we just go with standard methods?
Yeah.
Or
Yeah, so let me take this as one example. I have briefly mentioned before, even this dose toxicity picture, which really looks very, not very sophisticated, but this type of looking at dose toxicity as a curve was only the first time it was published in our area in 1990, like they call it continuous reassessment methods, paper by John O'Quigley. So only 1990. And it took years and years to implement these or his specific methods, or maybe some variation thereof. And in our company, we only started on a regular basis in around 2005, using these type of model-based methods for this MTD finding. So the first paper came out in 1990. In 2005, we started using our own version on a more regular basis. And before that, we were using insufficient or like ineffective methods like the 3+3 designs I briefly mentioned. So yeah, it does take a lot of time to come up, to really implement new ideas. Although the cycles of innovation maybe are shortening, I think everyone agrees that we cannot just wait 15 years to come up with a new idea. And it's not the regulatory agencies, it's just, it's much easier to follow what my predecessor, the previous protocol did. And I just quote unquote copy and paste because everyone is working on a huge time constraints and it's easier to copy and paste an old protocol. It may be the clinical people, they don't necessarily like that. They like the 3+3 idea in this particular example. So many clinicians wouldn't like the idea of the of this particular design. So this is just one example out of several. So when I joined Novartis 2004, I think most of our phase 2 trials in analyzing different doses versus placebo, we would use multiple testing approaches. Like, is the highest dose versus placebo, is it significant or not? And then the next, if it's significant, we go to the next lower dose. So we would perform a hierarchical sequence of t-tests. That was the standard in 2004. Nowadays, we use model-based dose response analysis in phase 2 throughout. So it's a big change, so to speak. Yeah. So there's another example.
thank you.
Okay, thanks so much. We're out of time, so let's thank Frank again for a brilliant presentation. And also like to thank Alison for organising the seminars at very short notice. So thank you, Alison. Okay, thank you all and have a good evening.